review paper on hormesis that was really interesting. And you've got this really interesting table in there where you talk about different drugs that seem to have, at least at certain doses, have neuroprotective effects that might be able to mimic, you know, the hormesis like effects of things like fasting and stuff. And so this is a really interesting idea. And I want to ask you about some more of these drugs. So you just told us the example of 2,4-dinitrophenol. It actually did help people lose weight. We know the mechanism by which it was doing that, and it had to do with the mitochondrial uncoupling stuff you were just talking about. But of course, you then said you could have too much of a good thing, and people started overdosing on it. The other type of drug I want to ask you about, because you have it in this table here, and it's also similar, I think, to the ones that are in the news a lot right now, are these GLP-1 weight loss drugs. So can you tell, is there anything interesting going on there? And are there any potential words of caution you might have, given what you just told us about the other weight loss drug?
So one quick comment before the GLP-1 analogs. Okay, so back in the 1990s, we did these studies with it's called 2-deoxyglucose. So it's a molecule very similar to glucose, but cells cannot use it to produce ATP. And anyway, the bottom line is it competes for glucose in the cell. So if you feed it to an animal, the cells will think they aren't getting enough glucose or as much glucose as they had been. And in fact, if you give it to animals, it will cause them to start producing ketones. It tricks the whole system into thinking that there's not enough glucose around. And so we found that 2-deoxyglucose was beneficial in our animal models of stroke and Parkinson's disease anyway. Okay, so for the GLP-1 analogs, so this is an interesting story, and I know a lot about it because, so I was a laboratory chief at the National Institute on Aging for 20 years. And so I had a big neuroscience lab with about 50 people in it or more. And one of the investigators in another laboratory, Josephine Egan, who focused on diabetes, mainly from a clinical standpoint, she developed a GLP-1 analog. That is a protein that's similar to GLP-1, but its amino acid sequence is a little different. What is GLP-1? Glucagon-like peptide 1. It's a hormone. It's produced by cells in your gut. It's released into the bloodstream when you eat a meal. And in particular, glucose seems to be the most important stimulator. So it's released into the blood. It does two things that are good for glucose regulation. Well, fairly directly. One is it stimulates insulin production by the beta cells in the pancreas. So when GLP-1 goes into the blood, that it's playing an important role in that rise in insulin when you eat a meal. So the insulin's function is to stimulate cells so that they remove glucose from the blood. Muscle cells, liver cells, brain cells, whatever. Okay. Second way that GLP-1 improves glucose regulation, it actually increases the sensitivity of cells to insulin so that it takes less insulin to stimulate removal of glucose from the blood.
I see.
Okay. So then at NIH,
so is that does that mean it improves insulin sensitivity?
yes,
Okay.
it does.
Okay.
Yep. Acts on your muscle cells, liver cells, even your brain cell to do that. Now, GLP-1 itself, it doesn't stay around in the blood very long. The reason is there's an enzyme, it's called a protease. It cleaves the GLP-1 protein. And when that enzyme cleaves it, it's no longer able to act on cells.
Mm-hmm. So your body releases it naturally after meals that are high in glucose. This thing improves insulin sensitivity, but it gets enzymatically broken down quickly. So it's not sort of meant to stick around for very long.
Right. And so these GLP-1 analogs, essentially what they did is they changed the amino acids in the region of the protein where the enzyme cleaves.
So it can't be cleaved.
It can be cleaved. So it stays around hours. And most of these are given by injection. There's even a once weekly, which is real nice for people with type 2 diabetes, right? Just subcutaneous once a week. That's it.
Yeah. And so what other effects does GLP-1 have? Because I would imagine, because I don't know all of the biology here, but presumably it does other things. And are there any potential downsides or hazards that people should think about here? Especially given the history of, you told us about the 2,4-dinitrophenol drug earlier. There's been other weight loss drugs in the past that act on cannabinoid receptors, and they help people lose weight. But then later on, we learn, X, Y, or Z goes wrong, and they have to pull it from the market.
Yeah, well, all I can say there is we'll see what happens. It has been used in diabetes patients for a decade.
I see.
Yeah.
I see. So it has already been used for diabetes per se, and it's now sort of been rebranded or co-opted into the weight loss area.
Yeah, and the reason is a third action of GLP-1, and that's on neurons in the hypothalamus that control appetite. GLP-1 suppresses appetite.
Which neurons is this acting on?
Oh my gosh. Would it be so is it AGR, which is it?
AGRP is the hunger promoting neurons.
Okay, so then it's the other ones.
It's acting on POMC neurons.
POMC, yeah.
Okay, so it's probably stimulating POMC neurons.
Yes.
I see. Okay.
Yeah. And so okay, so that's how it's acting. It isn't really acting in a adaptive stress response. I wouldn't consider it like an adaptive stress response mechanism. On the other hand, we started to look at the effects of GLP-1 and the first analog that was made at NIA called exenatide or exendin-4 on neurons, first in culture and then in animals, and found that it did have direct neuroprotective effects. In fact, based on work at Nigel Gregg, who was in my laboratory then, there have been 2 clinical trials in patients with Parkinson's disease of GLP-1 analog, and with promising results. They're doing a phase 3 trial. So I don't know about long-term, adverse effects. For example, I don't think there's been much at all done in terms of cancers. So anyway, so far it looks fairly safe, but I'm not promoting it. I promote exercise, eat a healthy diet composition, moderation in calorie intake, maybe intermittent fasting. I think sauna is good. I think the cold exposure is fine. You and I think are on similar wavelength. When you start tweaking with some hormonal signaling pathway like that.
There can be unintended consequences. And oftentimes the biology is incompletely understood. We understand one piece of it, but we don't even realize some larger picture it belongs to. Like, for example, naturally, as given my scientific background, like when I look at these GLP-1 agonists, you mentioned they have a specific protein domain that makes them rapidly cleaved by proteases. that evolved for some reason.
Yeah.
That's not a design flaw.
Yeah.
And so I would imagine that keeping these things around, constitutively, or at least for hours at a time, is probably eventually going to do something else that we don't even know about yet.
Yeah.